of acceptor, efficiency, completely directly, and it's a clear advantage of threat, as the variation of the lifetime of the donor in presence of acceptor. If you have a bimolecular interaction between two proteins of interest, you may describe this fluorescent decay as a B exponential, with the first exponential, which is the fluorescence of the donor alone, corresponding to the
about artificial intelligence for biological imaging. The comic talk will be given by Diana Mateusz. Diana is originally... Ouais. Pour les questions, en fait, il y en a un deuxième et puis on... Et de toute façon, vous êtes là... Et comment ça, du souffle, ça fait du bruit dans le... Ah, et si je parle, ça fait du bruit aussi ou...
On est bon? Bonjour à tous. Nous allons vous présenter. Ouais, mais bon, c'est. Tu l'entends, toi? Ouais, mais ça reste ce qu'on a. Ouais, mais méditerranée, c'est pas pareil, c'est pas la même table. Bonjour à tous. Bonjour à toutes. C'est bon là, toi, le son ou pas? Ouais, mais pas dramatique.
Ok.
Faites gaffe parce qu'en fait, vu que ça se sépare en deux, vous êtes tombé en plein milieu.
En fait, je suis arrivée, mais quand je suis arrivée, apparemment, c'est pour ça aussi.
Bah,
Et
en fait,
j'étais très contente de cela, d'avoir intégré le midi, parce que comme ça, ils sont...
Lyon est un mini-fold. Il y a des recoins, il y a
oui, non mais en fait,
c'est indépendant par rapport à tout...
quand ça fait partie, j'ai hésité à aller à l'INMT.
NVD3. Oui. Oui, parce que moi... Ouais. D'accord. Ah oui, il y a cette tendance-là. Okay. Shall we start? I don't know. Il n'y a pas beaucoup de monde là. Par rapport à l'audience. Moi j'essaie d'initier en fait la discussion entre toutes les plateformes. Alors, on en a vu, on y a réfléchi, mais le problème de la mailing list, c'est que ça se noie et puis en fait ça noie les gens dans la mailing list. Et donc du coup, j'ai fait faire un forum pour le public, un forum de discussion, partage. Par contre, il faut se connecter, mais je pense que Et après, le problème, c'est que pour l'instant, il faut que je recrute des gens pour y aller parce que j'ai écrit un truc, mais je sais pas. Il y a Jacques Brocard qui a regardé, il a dit ouais, c'est super. Voilà. D'accord. Eh ben oui, et là, c'est nickel, parfait. Parce que du coup, l'idée avec le forum, ça serait de pouvoir mettre un endroit où les gens peuvent piocher, discuter, parce qu'il y a aussi il y a une communauté plutôt vasculaire, une communauté plutôt neuro, qui viennent tous utiliser les mêmes outils, mais après ils discutent pas entre eux. Et en fait, il y a des outils. Voilà. Comme ils viennent, ils repartent sur les coordonnées, il y en a quelques-uns qui sont en train de faire. Non, non, c'est ça. Non, d'accord. Du coup, je lance dans... Ok. Alors.
Okay. Good afternoon, ladies and gentlemen. Welcome to the third and final session from the Module 3 about artificial intelligence for biological imaging. The coming talk will be given by Diana Mateus. Diana is originally from Colombia, where she completed her bachelor's degree in electronics engineering at the Javeriana University in Bogota. She moved to Toulouse for her master's degree in automation system and robotics at the LAS lab. She pursued with her PhD at the perception team from INRIA in Grenoble, defended in 2008. Her thesis focused on the registration and segmentation of articulated shapes using geometric interpretation of spectral embedding methods. After that, she worked for 8 years as a research scientist at the Technische University of Munich at the chair of for computer-aided medical procedure, where she received the prize Laura Bassi Outstanding Woman in Science. During this period, she was also partly a researcher at the Elmhorst Centrum in the context of a joint research group between the Institute of Computational Biology. And since 2017, she is full professor at Centrale Nantes and the LS2N lab in France, where she leads the multidisciplinary chair entitled MILCOM, standing for Multimodal Imaging and Learning for Computational-Based Medicine. The project focuses on the design of machine learnings to improve the acquisition and analysis of medical images in the context of nuclear medicine and ultrasound imaging. So please now listen to Diana's talk entitled Deep Learning with the Medical Images, Learning with the Small Data Sets and Few Annotation.
Thank you, Marine, for the introduction. So, as you have heard, I'm going to talk about deep learning. So I put here in small biomedical, because as you might have heard, my focus is more on medical imaging. But I won't talk about today about the why of these applications. I will just talk about how, in this context, we deal with learning, so deep learning with small data sets and few annotations. So what I have to say is that even if my examples come from a different domain, most of them, I would say all of them, are actually generalizable to other type of modalities. So I hope it's still useful for you. So I'm going to talk about mainly about two problems. The first one is classification, so classification with neural networks, where you get an image as input and a vector of probabilities as output. And the second one is segmentation, where basically you rely on this type of architectures where you get as input an image, and as output you get a label, but for each pixel of your image. So these two are the problems I'm going to be talking about today. So we know that for computer vision, these two problems are have been treated a lot, and we have very good results. Now what happens when we go to medical imaging? We will have a series of challenges. So from the collection of the data, we have to preserve privacy. We have some ethical issues here, so we have to be sure that we meet some ethical standards. Then from the acquisition side, which is I think something that you are also confronted with, is whenever we go from one hospital to another, if we have different machines, our images might look different. If we use different protocols of acquisition, they may also have different outcomes. This one is a big one, and one of the things I'm going to focus today is what happens in learning is that you, along with your images, you will also have to provide some annotations, some expert data, and especially in medicine, this is very costly. On the top, different to computer vision, our data might be 3D or temporal, which imposes some memory constraints. And then even if you get a very good result, it doesn't mean that your doctor will be happy with it. He wants to know why. So let's see if we can improve some of these challenges today. So I will be talking about these three applications. One is fracture detection, another one is mammogram classification, and another one is organ segmentation. So here I'll be talking about two different projects. And since I promised not to talk about why, if you want to know why, I will tell you later. But what I will focus is on the type of data we have. So here we have 2D images, 2D, and here these two applications are in 3D. And here we have what I call few data in medical. So as compared to the databases of computer vision, this is quite small. So we have around 1,300 images. Here we have a database with 410 and 2,500. And here is where we go very few data. So here we have 44 patients, and here we have 40 patients. And from these, we still want to do deep learning with this. In terms of annotation, so this data will be fully annotated. Here, they are annotated also, but only for the classification task. And what I will be talking about is also doing segmentation, only from these image-wise annotations. Here, I will have only partial annotations, and in this case, I won't have any annotations for doing segmentations in terms of the target domain. So that's the topic. And the common line of all the methods I'm going to talk about is how we add prior knowledge in order to constrain the difficult learning problem when we have few data and few annotations. Yeah, thanks. So for the fracture detection, this is a collaborative work with the Technical University of Munich and Pompeu Fabra. Here our classification is among 6 classes plus normal, so we want to say if it belongs to one of these classes or if there's no fracture. And we started as usual, just doing a classical supervised learning. We obtained some results. We said maybe this is not enough. So we asked our doctors to annotate regions, and by doing that we improved a little bit our scores, and then we try to replace that with a supervised localization tool. And then you proposed something that is called curriculum learning, and it's saying, okay, maybe we should train these networks by as we humans learn. So we should maybe start with the easy concept. So instead of just giving randomly the data to the network, maybe we could start with the easy examples, and then progressively adding slightly more difficult cases until at some point giving all of the examples. So what is easy and what is difficult? That is the key question of curriculum learning. So what we are going to use is the challenges that we had before. So maybe difficult is when we don't have many examples. This could be one hypothesis of difficult. Another could be difficult is where experts don't agree. And we have also asked, so this is a study that we did with 3 experts, and this is actually asking the doctors, what do you think is the more difficult class? And they gave us a ranking of these classes. So what changes when we do curriculum learning? In the standard conventional training, imagine I have only 12 images for my training. I have given some indices to these images. Before each epoch, your favorite PyTorch, Keras, or whatever is going to do for you automatically a random permutation, and then it will split your data set in batches, and you optimize for each batch. Now, if we want to do curriculum learning, what we have to change is that along with the images, we are going to also associate a probability that I have color-coded in this diagram. So light colors means this is an easy example, and dark colors means this is a difficult example. And I'm going to give this information coded as a probability, so that I can sample now using this probability. So I can sample first some easy examples, and then progressively I'm going to update that probability such that at the end I can also integrate the difficult samples. So in practice, how do I come from here to probabilities? So what I want at the end, this is what I want. For each class, I'm going to give a probability to all of the examples that belong to this class. So in my baseline, I would give the same probability to every class. Every class should be sampled with the same probability. And now what I'm going to integrate is these other 3 measurements of probability. So the first one is the one that I'm talking about, the class imbalance. So here I'm just going to say the normal is the easiest, because I have many examples of the normal. And then this one is the one with the smallest probability of being sampled in the beginning, because I have very few samples. In the second case, so this is the one that comes from the class imbalance. In the second case is where I ask the doctors to rank these classes. So I'm just going to convert the ranking with a very simple equation. I'm going to convert the ranking in probabilities. So the last one is using the inter-expert variability. And at the end, as you see, always I will have just a value of probability for all of the examples in that class. So each sample gets assigned the probability of its class. Okay, so that's the technical part. And how do we know if it actually changes something for my problem? So what you want to do is to compare the results when you train with a curriculum. So when you go from easy to hard, to the results when you train with the anti-curriculum, meaning that you start training your network with the hard examples and then you go to easy. And if you see a difference, then actually it means that your prior knowledge that you integrated through this curriculum is actually going to help your classification. So let's see if this helps. This is what happens if you just use the normal random sampling from your favorite framework. This is if I sample equally from each class, uniform distribution. This is what happens if I use the frequency, so the class imbalance. So here, this is an F1 measure. I want the scores to be high. So what we see here is that my results actually dropped. So this didn't help at all. And I will tell you later why. But this is what happens if we include the results of the expert ranking. So how the expert thinks which classes are the easiest and which classes are the more difficult. And similar if we include the results of the inter-expert variability. So here you see this big gap between the curriculum and the anti-curriculum, and here from the curriculum and the anti-curriculum, from the expert variability and expert prior knowledge of difficulty. So what we think happened here is that it is not because you have few examples of a class that this class is difficult to classify. Some classes that you have very few samples actually were from very big fractures where the bones separate a lot, and then this actually is quite easy to split even if you have only few samples. So what we get from here, we have two curriculums that actually helped and one that didn't. And the question is, what happens if I don't have any prior knowledge? So one of the things is, of course, if you have an expert, you can ask your expert. This will be quite easy, but even if no expert knows what is more difficult and what is easier. So we also extended this to another method in which we had an observation similar to one example that a speaker talked about this morning, in which we have predictions at different scales give you different results. But what we remarked is that when they are giving you different results, usually there is a misclassification. So it's actually an indicator that probably if your network is giving you different predictions at different scales, it's probably because it didn't learn very well what happened with this example. So we want to claim that this disagreement is informative, and we want to learn from this system uncertainties. To do that, we are going to rely on dropout. So if you have followed, I probably hope David introduced this. No? Okay. So in the dropout, what is going to happen is you have your network, and during training, you will shut down some of the neurons randomly, and then you will let the backpropagation flow to the active neurons only. And then you will shut other neurons in the next iteration and do the same, and shut another set of neurons in the next iteration, and so on. So the idea here is that this will prevent your network from overfitting. So if all the things were focusing on a single neuron, if you shut it down, then you will let the other neurons learn this time. And this is used typically for training by default, but what we are going to use is dropout for in test time. So it's the same principle, but now what is going to happen is that when you have a series of networks of neurons that are shut down, you will make a prediction. This will make you P1. If you shut another series of neurons, you will get a second prediction, so a different prediction, a different prediction, and a different prediction. And what we're going to see is if there is a lot of variability in these predictions, it means that probably your model is highly uncertain of this example. So it gives you an idea of if it's difficult or not for the network. Okay, so this time we don't have prior knowledge, but we are going to see how the model responds from different dropout outcomes. And if there is a lot of variability, we will say that the example is actually difficult for the network. And there is many ways in which we can compute from these different examples the uncertainty, and one we use is just averaging the predictions and computing the entropy. So this is actually 2 lines of code. There's nothing complicated here. So just to recap, before what we had was a series of fixed probabilities per class. So all of the examples that were in my normal class were getting exactly the same probability. Now here I have some probabilities per samples, really how each of these examples is behaving when I shut down some neurons. So if it's changing a lot the prediction, then I will say that it's difficult. And if all the predictions with different dropout examples are getting the same result, then I will say this is easy. Okay? And of course I can update this as the training goes, so it's going to be evolving over time. This is called self-paced learning. So if we combine the two, I have a series of data and have either prior knowledge or uncertainty to define a scoring function such that each of my sample gets a high score or a low score. And if before I mentioned that we could reorder and feed the network with a different order each time. There are also some strategies that we explored, like subsets. Maybe you start training with a fewer amount of points, not all of the points, and maybe you can also just weight the use this scoring as weights, similar to boosting for those that have done more classical learning. Okay, so what do we learn from this? In the left-hand side, you see what we had before. This is exactly the same results as in the original case. So we have here prior knowledge, and I have just added other types of pacing methods. Usually all of them help. This is the 3 class, which is easier, so there is no big gain. Or usually the gain, it's in the more difficult 7 classes. And this is the comparison when we do this without prior knowledge, but instead look at the uncertainty. And you see that also it helps, so all of the results get a little bit improved in the 3 class, but they get a big improvement in the 7 class. So the conclusion here is if you don't have prior knowledge, maybe you can also just look at your network, how it behaves for different dropout instances, and this could also help you get some improvement. And we did a little test with MNIST here. So we wanted to check if it's actually our claim of our claim that curriculum learning helps with few data was true. And so we did this test with a control data where we have a lot of images in NIST, but here this is error rate, so we want to be the smallest is the best. And what we trained here is the same thing. So on the left we have prior knowledge, on the right we have uncertainty. And what we have here is 30% of the data, 50% of the data, and 100% of the data. So ideally, if you are using just classical learning, you will end up with a high error. And if you use any of the techniques that I have shown here, you will make your error drop by including some curriculum. And this is, of course, mostly noticeable when we have smaller amount of data. We also did some tests for class imbalance and label noise with similar results. And if I conclude on the curriculum learning part, then if you have a multi-class classification problem where you're stuck with your scores, this is maybe a good solution that you can implement. It doesn't change at all the learning part. You only have to change the way you feed the data, so it's really at the beginning. And you can use either prior knowledge or model uncertainty to do this. So it has a very easy integration. Okay, so maybe I move to the second problem that I want to deal with. This is mammogram classification. This is a collaboration with the company Hermi. And here we want to do a classification of images into normal or abnormal. So once more, if you just train your classical CNN, you will end up having some score, and the doctor comes to you and say, yeah, but why? Now it made a mistake, so how do I know if I should trust or not this algorithm? There had been a lot of methods trying to do something like class activation maps, so they tried to, instead of flattening all of your last layers of the network, they tried to show you some heat maps that go between blue and red and tell you, okay, maybe the red regions is where the network look at for making this decision. The problem with these class activation maps is that they are very low resolution. Now there are some that are now also becoming higher resolution. And what we want to do is instead learn to decompose the images into abnormal and normal regions. So this is my input image, and I want to be able to decompose it, so disentangle the information into a part that is going to be the normal part and another part that is going to be the abnormal part. And why I'm denoting here in red, in pink and blue are the regions that are annotated, but I'm not going to use them for training. This is only for validation. So this is one of my few examples that have been annotated and where I can actually compare for evaluation. But as input to my network, I only have image-wise labels. So I know if this full image is normal or abnormal, and that's it. I don't know where the abnormality is. And I actually want to be able to separate the abnormal part just from image-wise labels. This is called weakly supervised segmentation. So how do I bring prior knowledge here? Similar to other examples that we saw this morning, I can rely actually on simulation. I know how my lesions look like. So my lesions might look like masses, they might look like distortions, or they might look like clusters. And what is good about mammography is that we have many, many normal images, because women go through screening regularly. So we can collect those images, augment them with this type of simulated lesions, and try to learn an encoder-decoder architecture where we can separate the two things. So this is supervised, but it's supervised only with knowledge of how to simulate the lesions. So here I haven't done any expert annotations, it's only I have integrated knowledge about how to simulate my data. So now, and this is just practically the loss that we use. So we just compare the sum of the two channels in order to do a reconstruction loss. So now I can actually combine the two things. So the class activation maps with the abnormal separation, they bring complementary information such that I can now explain if really one of the images is classified as malignant. I'm able to say for sure that it comes only from the abnormal part. So this is going to give some explainability to the doctors. So I had my class activation maps. I'm going to combine the two things. From the topper side, I'm going to split the data into the normal and abnormal part. I'm going to use the abnormal part, and I'm going to combine it with the class activation maps to decide whether it's malignant or not. And I'm going to do this actually during learning. So I'm going to combine the losses, the reconstruction loss from here and the classification loss from here. But so this is already giving me some explainability. Now I want to get better results. So I bring more prior knowledge here. More prior knowledge, for instance, if I know the size of the lesions, I can actually add a loss. So maybe in your cases, you know the sizes of the cells or the sizes of the elements that you want to segment. So you can add a loss telling what is the actual range of possible sizes that you think are admissible for the object that you want to segment. And then you penalize anything that is above or below this size. And you can also use things that maybe you are commonly using in post-processing. So morphological losses that we usually use, for instance, if we know that we don't have a lot of isolated points, but maybe we have only fewer convex regions, we usually use something like closing or a top-up operation if we want to give priority to bigger regions. But we want to integrate them, and we show how to integrate this type of operations directly into the training of the loss, not as a post-processing operation, but during training, so that once you have learned with it, now your outcome, you don't have to post-process it anymore with this type of operations. So we add that also here, these two losses. And then if we recap, then the learning goes in two steps. The stage one is this self-supervised pre-training with the simulated data, and in the stage two, I'm going to use the multitask learning with these three losses to further constrain the problem and bring my few examples of negative, malign cases inside the network. So this is some example of how you can control the outcome of your segmentation. So these segmentations are coming for free, I insist. I don't have any expert annotations used during training to get these results. I only have the image classified as benign or malign, that's all. And here is different combination of the losses that I have showed you before, so the size, the morphological, and so on. And this is how the results of the segmentation look like when you combine some of them. So in our case, we prefer this option, even if you get some false positives because overall over our data set it was the best performing one. So what we learned from this example is that we can actually get similar performance in classification, localization, and segmentation to fully supervised training. So we compared to some algorithms where they were using expert annotations during training. And maybe for your case, what is important here is this prior knowledge. So if you have way to simulate even roughly your data, this will really help a lot in getting better results. And if you know the size, the number, or other prior knowledge, you can incorporate that into the loss to better constrain the outcome of the segmentation. And even if you don't have prior knowledge, you have seen already in some other examples this morning that some other restoration from perturbation pre-training is also very helpful. So this is from an example of another paper. Okay, now I have 2 left. Let's see if I can manage. How much time do I have left? Perfect. Okay, so these are the most dramatic in terms of number of examples. The first one is going to be about muscle segmentation. We have this type of data, so you see that we are not able to image a full muscle with ultrasound at once, so we need several sweeps that we record by tracking a probe in order to reconstruct this type of data, and this is what we want. From this, we want to segment the different muscles of the of a person. So this is the acquisition. There are some manual annotations, so you see they are actually annotating the images on the partial views, not on the full views. This is the type of annotations that we could get. Then they also make several acquisitions with different parameters of their, and we might need to use techniques like the Perrine was presenting this morning, where we have to register between different parameter settings of the machine in order to get all the labels in the same coordinate system, and then at the end we are able to get some examples from which we can learn. So these are propagated from here. So when we learn in this data set, we tried 2D unit, it didn't work, it was not enough, so some slices were very easy to segment, but then the next slice you will get bad results. 3D unit, it does not fit in memory. Our volumes are 1,400 slices across, and then you have 512 times 512 per slice. So if you want to put this in memory, then it becomes a bit complicated. And then we also have this problem of partial annotation. So what we are going to do is instead we are going to treat our volume with sliding volume approach. So this, I'm going to take, let's say here, 3 contiguous slices, and then I'm going to process it progressively as in depth. And this will be also useful if you have spatio-temporal data. So here, this depth could be also time. And what we are going to use is, we are not going to use only prior knowledge from this spatio-temporal continuity in our data, so we know that the muscles won't abruptly drop from being there to not being there, so they will smoothly move in our images. But also our labels should be spatio-temporal coherent. So when we used this 2D unit, and I mentioned that the results in the first slide were very good, and then in the next one it was dropping, this is a little stupid. I mean, if the previous it was doing a good job, why is it not doing it now? So what we want to be sure here is that if you get some good labels at some point, that you are able to propagate this knowledge to the contiguous slices. So if you have spatio-temporal data, you might have heard of long short-term memory networks. These are networks that incorporate temporal knowledge, so they have gated gates inside that allow you to process stages of your data. And this bidirectional means that I go from the first slice to the last one, and then I do the same procedure from the back to the front. So I have here a typical encoder, decoder architecture, but in the middle we have added this bidirectional CLSTM. Okay, this is okay, but it doesn't give us the best results. And sometimes what we would wish that we could do is correct the results of a network. So we want our network also to be interactive, such that if I, at some point, know that these results are not so good, maybe I could provide some annotations and correct this portion of the network, okay? So here I have some information that is intensity. Here I have some binary labels. So these two are each of them going to focus on the features of the two different incomes. And then we have some global matching between the two so that they can talk to each other. We have, again, our temporal model, the decoder, and so on. But this is not enough. I mean, there is something that is a little bit difficult, is that what happens with this input if I don't have anything inside? So how do I train it, right? So what we are going to do is that we are going to alternate. If we have some input annotations that we want to give to the network, we feed them here. And if we don't have them, then we are going to look at what was the last estimated masks that we had, and we are going to feed them. So we are going to try to propagate them progressively by give them as input to the network. So this is because of the 2D unit example that I talked. So if you predicted something that is good, maybe it should help the network to decide that the next predictions should be also similar to what you predicted. So we have some propagation of these labels. So that's why we call our network interactive few-shot Siamese network for faster muscle segmentation and propagation volumetric ultrasound. So here few-shot is because we really have very few volumes to learn from. And there are some other tricks for the loss and the global matching model which make a little difference, but these are not the key of this work. So what do we get? Here is the supervised setting, so just using bidirectional CLSTM, and the rest is off. And our results are on the pink. So you see here is error, so you want this to be low. And pink does much better than trying to slide window with a 3D unit or a depth reading or a VNet. So temporal encoding, CLSTM works if you have spatiotemporal data. And then if you have really very few data and you want to incorporate some annotations from the user, so if we have all of our models activated, here when we called interactive settings, so we have simulated the fact that we give 3 annotations in the beginning, 3 annotations at the end, and 3 annotations in the middle to do these results. And then the rest is pure propagation, okay? So we have very few annotations. And what we get here in that case is the yellow case. The results are also having a very low error. So recurrence also helps. And then there are some visual visual results. What it's good here, then it's something that adapts online. So if you include a correction, you are able to make new predictions with this correction. And this allows us to go from 5 hours of initial segmentation that it took somebody to annotate this data to just a few minutes. And if I go to the last one, so the last one is, I have, okay, it's perfect. So the last one is about unsupervised domain adaptation. So here, imagine you have a modality and you have some ground truth and you train a neural network to make predictions in a supervised setting. This is classical, this is what we do all the time. Now what happens if you know that exactly the same thing that you're going to segment can be imaged with another modality here, CT for instance, and you pass it through the same network without training it on this data, the prediction will be bad because you have not seen this type of data, right? And if you compare to the ground truth, of course, there will be a big drop in the performance. So this problem is called unsupervised domain adaptation. We want to be able to segment this image by only having annotations in one modality, and we want to segment in other modality when we know the two things are imaging the same structure. So this is a domain shift, and we want to correct that domain shift. So here is the same problem. We have data and annotations for MRI, and we want to know how can we bring unlabeled data into the training stage such that later I can use this model to segment images from the second modality. So there have been works using GAN stylization. The problem here is that they have many parameters and they may generate non-plausible data, so they may generate things here that do not resemble heart. There have been also some methods that use variational autoencoders. So variational autoencoders are nice because they constrain the latent space, so they really ensure that whatever you sample from them is plausible. So they stick to the region of the latent space that they know. They look something like this. So instead of just learning a dot at each in the low-dimensional representation of your network, you are actually trying to learn a distribution, usually a very simple distribution, a Gaussian normal distribution. And then you try to recover the output by sampling from this distribution. So here, the good thing is that because you are sampling from a distribution, then you cannot sample from here because you have never seen. In GANs, you can. So that's the good thing about variational autoencoders. The bad thing is that when you use them like this, it's a little too much, all these connections. So these connections are very complex composition, and they are using also variational autoencoders to recover the intensities. So we would like to make a little bit simpler representation. This is another example where to solve this unsupervised domain adaptation, they just used a unit, but they used optimal transport here to match the distributions of the data, and here too. And we want to get inspirations from these two later type of works to solve our problem. So why optimal transport? This is something that is quite trendy in these days. So here is an example of optimal transport for classification. You imagine you have your target data set and your source data set. So here you have supervised annotations, and you train a classifier, this green line. What you want to do in unsupervised domain adaptation for classification is you want to bring this target and the source to the same space, such that you can use the classification model that you trained for solving the problem. And we want to extend this for segmentation. Okay, so we want to use optimal transport and variational autoencoders. Here is what happens if we just use a variational autoencoders when we feed from two modalities. This is going to be your latent space reduced using a TSNE visualization, so going from 128 to 2 dimensions. This is what they were using, trying to recover the intensity. What we want to say is maybe intensity is too much. Let's focus on the task that we want to solve. Let's focus on modeling in this low dimensional space only the segmentation. So we are going to go to the encoder, decoder. This is just to say that we are learning a normal distribution here. And when we sample from it, we are going to obtain a segmentation map. This is what we had originally. When we do optimal alignment, we align our two Gaussian distributions very well. And then we sample from this, and we are able to obtain CT segmentations with the same model, only training with MR data. So this is the type of results we get. So this is my CT slice, which the model has not been, it hasn't used this ground truth. It uses this as input, but it has never seen this type of ground truth. Here is what we would get if we train this with a unit, for instance, supervised. So these two models see the ground truth, and these three models do not see this ground truth. Here is our result. When we feed 16 volumes of CT to this network without ground truths, we are able at the output of our network to get this type of segmentation for a modality that we have never seen ground truth for. So we just rely on the ground truth from MRI. Okay, So I have some more results on that, but let's wrap up. So I hope I have convinced you that we have similar problems in medical imaging with microscopic imaging. And I hope also that I have convinced you that bringing prior knowledge, either in the terms of difficulty, so of the task, or shape and size, or spatio-temporal consistency on bringing prior knowledge from other modalities could help us in solving these tasks. So that's all from my side.
Okay, if you have any questions, well.
Thank you. Very nice presentation. I have a question about the first part of your presentation. So you use prior knowledge or model uncertainty with your dropout strategy to improve the results. Do you think that your model uncertainty actually recapitulates the complexity of your data, like the prior knowledge would help? Or is it something different, in which case you could combine the two to get even better results?
We tried, it didn't work. But I mean, this is something that we are hoping that we can manage to find a way to solve them too. But at some point, it just didn't help in the convergence, combining the two.
Thank you a lot for your presentation as well. Still on the first part, I just have two questions actually. One is regarding the validation itself, because you have such inter-operator variability on the annotation. How do you estimate the F1 score for the one where you're not?
So usually what happens is that the very experienced raters, you cannot tell them they are not right. So you have to accept. But the annotations from lower, so from interns or early stage doctors are reviewed by the expert. So there are some other cases in which you would like them to sit and decide together. But in this case, the ego of the first one wouldn't allow this. So we stick to the simple, more expert has it right.
So at the end, you have some kind of ground truth for this one.
Yeah, I mean, it's still noisy. Whenever you use expert annotations, you can never say it's ground truth.
And one short question following it. Could you imagine then that instead of the annotation done by expert or young doctor, whatever, you could use the result from another classical method, which would be noisy and don't give a very good result, in order to train on not having to annotate anymore then?
If we manage to do this would be great, but I'm a bit skeptical that it would work. So the task, the doctors need 10 years to do this right. So.
I thank for the nice talk. I want to ask a question about the second part of your talk. I really like the idea where you kind of highlight the areas where kind of network learned, because it's kind of like, you know, it's black box, but now you kind of know what is happening. So I have two questions. The first is like, did you try to apply to your first data set with the fractures, just to see like if it really sees fractures, or maybe, and how much this weak segmentation has like the, you know, what are the limits of this weak segmentation? Because it's kind of also something a priori knowledge that you put in the model, but it not necessarily have to be there. I mean, in principle, it could be that, for example, the doctors always make picture of the fractures where the knee is, you know, like in a bit different angle, and this is what the network learns, but not really the fracture. So, okay, so these are two questions. Thank you.
So we did try with the simplest cases. So we used global average pooling and class activation map to see the surfaces, and we played with, I don't think I have the results here, but we did see some correlation, but we didn't push it to the same extent like the mammography, where this separation in the two channels is much more enforcing the causality of the prediction. So I think really this separation makes a big difference. Here you cannot just decide on the view, this will not work anymore. So here you will prevent this type of correlations that are not meaningful.
Any other questions or is okay? One and it's last.
Thank you for the presentation. I have perhaps a really naive question because I know nothing really about deep learning and so, but can you help the program by simplifying, in fact, the task for the fracture? For example, doing first segmentation to learn to him this is the pelvis and this is the leg, and then after making a decision, is it something wrong in the leg or in the pelvis? Because there you will have a program that is able to do diagnosis on several parts of the body, because he knows which part of the body is looking at.
Yeah, so actually one of the things that they wanted to do in the beginning was extend this to other parts of the body, and this was a big issue. We also considered having 3D models of the bone to create projection data from false CT data. Unfortunately, we didn't push this direction, and what is really difficult is, I mean, in this case, if we have really CT scans from which we can project data, the segmentation becomes easy, but otherwise doing this from 2D is more difficult than actually classifying, so it would not help in that direction.
Thank you.
Okay, thank you, Diana, and then you can well, thank you, her. While Zaf is getting ready, then I can introduce him. So it's a final presentation of the module 3. This will be given by Asaf Zaritsky. Asaf comes from Beersheba in the south of Israel, where he completed his bachelor's and master's degrees in the field of artificial intelligence in the Ben Gurion University Department of Computer Science. Then he pursued with his PhD in the field of computer vision at the School of Computer Science at Tel Aviv University. After a postdoc in computational cell biology at the Department of Bioinformatics and Cell Biology at the University of Texas Southwestern Medical Center, he came back to Beersheba as a PI of the Lab of Computational Cell Dynamics from the Ben Gurion University of Negev. He caught the interest in microscopy while watching a microscopic video of cells moving in a group during his PhD. As a computational scientist, he identified these motion patterns as an opportunity to make a significant difference in understanding complex dynamic biological systems. Oh, there is a... Okay. Ever since, Asaf's field of research is at the interface of cell biology and computer science and deals with the understandings of complicated processes in cells through a range of computational methods from the field of data science. With the help of advanced artificial intelligence tools, including, of course, deep learning now, his lab produces biological insights and dedicates analytic tools to reveal hidden patterns in dynamic cell imaging data. His current research questions is how do cells communicate to achieve efficient collective self-organization and dynamics in normal process and disease. Okay, please.
Okay, thanks for the kind and extensive introduction. Let's do it a little bit differently. If you have questions, then stop me and just ask, okay? This, I think it's nicer than just speaking all the time, especially after all the Zoom meetings that I had in the last Zoom presentation in the last several years, so couple of years. So, and yeah, and can you tell me like 10 minutes before? So yeah, okay, thanks. Okay, so so I'm going to tell you how to find hidden patterns in dynamic cell image data. And I'll start from the beginning. For many, many years now, we're staring under the microscope. We're seeing a phenotype, we're explaining it, and this is how we learn new biology. And it's not new already. It's already in the last several tens of years. We know that we also need to put numbers in order to show that what we find is real. So in order to do that, we have all these amazing tools that are built and developed. Here are a few examples of them that allow us to take our images and extract quantitative information and then measure our phenotypes and see what is real and reproducible. This is not a perk anymore to have these tools. It's not nice to have. With the amount of data that is generated today and with its complexity, and it's a necessity. We cannot anymore do manual annotations of data. We need automation to actually measure stuff within these huge amounts of data. But actually, it's more than that. Even in a small data set, you can think of patterns that are really hard to quantify. So if you think about it, Megan and I are proposing that there are two steps when we think about quantification or computer vision in cell imaging. One is the signal-driven processes that we try to do tracking, segmentation, registration, and this is the input to the higher level where the questions that we want to ask are driven by the biology. I think one of the cool, I think that most of the emphasis today in the bioimage analysis community is here, and I'll try to convince you that there are cool stuff here and we should put more, computational people should put more attention here. I think one of the coolest parts here is that in computer vision, the state of the art is what the human eye can capture and assess. So the ground truth is getting, the best performance is the human performance. But in cell biology, with complex dynamic processes, the eye cannot capture this information, and computers can sometimes do it better than the human eye. So for these processes, I propose to switch the order and first quantify, and then go back to, always we need to go back to the biology and try to understand that. Sometimes we can see that, sometimes we cannot. I'll try to show you several examples of that. In order to do that properly, of course, we need to understand very good understanding of the domain, of the microscopy techniques. What do we want to look for? Where do we want to look for it in an image? What techniques are we going to use to actually extract this information? So for the rest of the talk, I'm going to show you two examples that I hope that will convince you that this way of first measuring stuff and then going back to the biology and trying to understand it. Here will be I'll be able to show you to see something, and here the numbers, the eye can, at least my eyes cannot capture anything. It's all the insight comes from the numbers. Yeah, and in a session that is deep learning oriented, this part has no deep learning. So. Okay, so the first project is a project that I started while I did my postdoc at the Gaudens Danuser Lab at UT Southwestern Medical Center. My colleagues who collaborated with the main collaborators with Andrew, who joined me with the computational aspects, Andres, who did all the experiment and the mice work, and Eric, who established the analysis, the experimental system, and the questions that we eventually, I think the main contribution here is actually, we talked about deep learning. It's very powerful, but it's a black box. So we try to reverse engineer a deep neural network and to go into the network and try to understand what is captured there inside. So the biological question that we asked, or diagnostics, was we were fortunate to have access to a data from the Sean Morrison lab, where they xenographed melanoma stage 3 in patients. So they were extracted they were already metastasizing melanoma from lymph nodes. They were xenographed into mice, and the prognosis of the mice was correlative to the prognosis of the patient, which gave us a very powerful system to study melanoma metastatic aggressiveness from the mice model. There were two classes here, two labels. Of metastatics. There were some of the patients that the metastatic went to the lungs, which is considered low metastatic, and the other where the metastatic went all over the place, and this was considered high metastatic. So the Morrison lab, you see, this is an old paper. They tried for quite a few years to extract all kind of readouts to discriminate between these two, and it wasn't very successful. And oops, and we were hoping, that we can do some extract something from the morphology of the cells, the motility. So what we did, Eric established an assay where he took the cells from the mice and he placed them on a thick stab of collagen, and they were live imaged like that, just the face contrast images. And we measure all kind of stuff. We measure the morphology and motility and whatever you could think of, but we could not find any discrimination between high and low. So what we thought to do next was there was still a lot of texture in these images, a lot of stuff that was going on, and we were hopeful that the powerful, mighty black box will capture the information that will allow to distinguish between those two classes. So what we did, we developed an image analysis pipeline that identified and tracked single cells within these images, within this time lapse, and these were our atomic units of analysis for the rest of this project. So what we did here, we used the autoencoder. It's not this autoencoder, but this is a simplification. It was an adversarial autoencoder to encode the appearance of a melanoma cell. So luckily, we already heard the talks that showed us this type of architecture. So I could go quickly over that. We have here a part that is encoding. Here's the part that is decoding an image, and this is the compressed representation of an image. So this is an encoding that allows us to extract the full-blown image, and the optimization is minimizing the discrepancy between those two images. Importantly, this training process is completely unsupervised, so we don't tell the network anything about whether this melanoma came from a high or a low metastatic efficiency patient. Actually, we even gave to this network a lot more data. We gave it data from cell lines and melanocytes and clonal expansion of melanocytes. We gave it all the data that we have, we threw into this network to get a good representation of a melanoma cell. And now, after we had that, we used our encoder to extract a quantitative representation of the cell appearance. We plugged it into another classifier, and here we could distinguish very nicely between a low metastatic efficiency and high metastatic efficiency cells. We did it in a way that it was a patient independent, so we didn't show the network any data about other patient, and it was about the patient that is in test, and it was robust to all kinds of manipulations that we did for the images. So we did some blurring and added noise, etc., and it was pretty robust. Okay, so this is fantastic, and the next step, actually, we also validated that. So we went back to the cell lines. We had 6 cell lines that are considered highly metastatic. And what we did, we used our classifier. So here is a classifier for each patient. We had the classifiers that did not include data about this patient. So we ran our classifiers on these cell lines data. And we predicted that the A375 is going to be highly metastasized, and the MV3 is going to be much lower in terms of metastasis. And we validated that in the mice, and indeed, within the A375, we saw metastasis all over the place, and the MV3, we didn't see that. Okay, so it's great. I mean, it's still, the numbers are small. We didn't have tons of patients, and still, we need to validate that in order to say that we have something that is really useful. But at least it's, it's a start, and it's cool. But the question here, I mean, we want to understand, okay, so what? I mean, what did the network learn? What information did it use? We tried to measure all kinds of morphology features and motility, and nothing worked. So what did the network capture that we couldn't? So the first thing that we did, we have a classifier, so we can rank our cells by the classification score. So you can see here cells that the classifier thought that they are low metastasis, and here's a high metastasis. And I stared at thousands of these images, and practically I would say you cannot say anything about that by staring at these images. Why? Because the variability comes from so many places. I mean, it could, in the representation of the cells, there is so much that is not specific to the metastatic function. Why would we see it when ranking the cells when it's a complex phenotype? So yeah, this was a difficult point here. But then we had the idea of doing morphing. So the problem was that we had a lot of variability within the data, and we couldn't capture what was the specific parts within the feature space that was relevant for the task of the classifier. But what we could do, we could take a vector that represents a cell, and we could push it into the high metastatic domain or to the low metastatic domain, and this reduces all the or most of the non-relevant variability. So basically, we fix everything. We just push the vector of the same cell into a domain that the classifier thinks that it's more aggressive or less aggressive, and then we can look at an image as where we focus on the relevant parts within our variability. And here is what we got. Here we morph melanoma in silico, so you see a cell going from high to a low and from low to a high. The morphology changes are not really are not something that we captured within the live imaging. We don't think that it's real, but what you can see very clearly, there is a change within the brightness of the face of the in silico phase contrast images. This is one thing, and the second we found these mini protrusive extensions out of the cells. So we thought that these could be properties that are emergent cellular properties that are specific to the action of highly metastasis cells. Importantly, we couldn't use what in the previous talk we heard about the ideas of localizing parts for explainability. It's in medical imaging, a lot of time you look at the slide and you try to understand which are the regions that are specifically for the classification. Here we cannot do something like that because the properties are emergent properties within the whole cell level. Okay, so the next part, so this was also reproducible and it was specific, which I'm not going to show you the data for, but also I want to show you a nice validation that we did. So we looked for cells that spontaneously transitioned from low to the high. I mean, these are cells, they could be either low, either high, we don't care. I mean, we care that the classifier thinks that they are transitioning from a low to a high. And this means that we have less variability within our data because we're looking at the same cell. And now, this is the live imaging. And now when we stare at the images, we can validate, and we do it also for multiple cells, we can validate that it's the brightness, the change in the light scattering of a cell is a characteristic of the transition from a low to a high metastatic efficient cells. Why? I mean, we don't know. It could be a change in the cell stiffness. It could be something within the cytoplasmic composition or organelles organization within the cell. But it does give us a starting point. Now we can start look and understand and try to explore what are the properties that are specific. And we couldn't find, in live imaging, we couldn't find anything convincing about the mini protrusions. But we're not sure about that. There is some literature about small protrusions that are linked to survival with melanoma. And we think that maybe we cannot say it because it's too subtle, even within the live imaging, but who knows? Okay, so to summarize, we think that we used this type of in silico morphing to identify, to do reverse engineering for a neural network, and to amplify the specific properties that are driving driving, I would not say driving the metastatic efficiency, but driving the network to think to discriminate properly. So if you think about more generally, the idea is here that we are trying to think on how to reverse engineer, how to go into the neural network and try to understand what it actually learned, how it did its discrimination, and use it for something beyond being a tool for classification. Yeah. Okay, so this is, yes, look. So,
yeah, sorry. You mentioned, that scattering seems to be associated with this metastatic, activity. And my question was, could one imagine that this is because the cells that are metastatic tend to migrate more, Therefore, they need, you know, more cytoskeleton and also other kind of active proteins and essentially more dry mass to be able to just move, crawl around and move around.
So we couldn't find anything that relates to migration. It's not just in these images. We did all migration and invasion assays, and there is nothing there. I mean, we couldn't find anything that relates to that. Yeah, now I think we need to, I mean, it's not, I'm not going to do it. I'm completely dry lab. But if Gaudens wants to continue to actually explore that, he needs to do experiments that actually try now to screen, you know, to screen the different properties that yeah. And use this machinery to see, yeah, to make predictions. Yes.
Thanks a lot for the very interesting talk. So I remember that there was some interesting work by Gustavo Kundo Rode, who basically used a different metric to embed the space so that he could do something like a PCA, but the principal modes that he would get would be like morphing modes. So it would be, the first thing is a size change, the second thing is a stretch change, and so on. So it might be interesting to look whether you can combine sort of this feature vector representation with this morphing-like spatial embedding. I think they called it like a local embedded space or something.
Sorry. So I'm not aware of this work, or maybe I don't know. But I would say that I skipped the whole part where we did try to look at the feature vector, at the unsupervised, trying to do dimensionality reduction and visualization, and it didn't give us any information. There are several reasons to that. One of the main reasons here is that the variability, that the main axis of variability is not necessarily the variability that is relevant for the classification, for the classifier, right?
But what one could observe in your movies is that they didn't look 100% realistic in the intermediate, right?
Of course.
Whereas his movies somewhat looked all realistic because this embedding seemed to work nicer. And I think that might be a good way to pre-rotate your space, basically, right?
Okay, let's take it offline, okay?
Yeah, sure.
Thanks. Okay, I didn't finish. But if you have questions, then go ahead. No. I did want questions in the middle. I said that. So yeah. So go ahead.
No, so I was wondering about the pushing and the time. So you have time, but I missed somehow. Where did you encode time, or do you pass the volumes or just single images? And when you say you push, so when before you do the morphing, you said you push this in the feature space. I also technically, how is that done?
Okay, so in terms of time, the things that worked best for us is just taking the time average of everything, using single images and taking the time average. We tried all kind of tricks with temporal representations. Nothing really helped here. The cool thing here is that we can do is train the classifier on the average time, and then use it on single frames, right, when we go to the live imaging. So that's regarding how we use time. We just averaged. Regarding the second question was what? Yeah. Okay, so the main thing that we did, I mean, it was really, we were super lucky here. We had one feature within our representations that was correlated with the outcome, so we changed it. Later on, what we did, we expanded that to, so this is how we did it originally. Later on, we expanded that to more generally, so going along the gradient of the classifier score. Yeah. Yes. Yes, phase contrast. Rather what?
Basic.
Yeah, so it could, yeah, I mean, if we change, so first, I mean, it's basic, but it still has a lot of information within it, and now there are more and more papers that actually show, I'll even show something that there is a lot of information hidden in phase contrast. Yeah, if we have a better, yeah, you know, more quantitative phase contrast technique, it might be better. Yeah. Okay. So we're now trying to, so now in my own lab, we're trying to expand these ideas to in vitro fertilization, to expand the technique and implement it to in vitro fertilization. First paper on this topic from Itai Ehrlich. It's not on the topic, it's actually more on the topic of using noisy labels and information. So it's actually a little bit related to the last talk. Is already out, and Oded is trying to implement what I showed you on the melanoma on a more complex system of in vitro fertilization. Regarding information within phase contrast images or bright field images, recently, actually it's not very recently, but it was shown that you can take bright field images and predict, you know, you need to be very careful about it, and you cannot say anything conclusive about every organelle, but you can predict some of the localization of some of the organelles within the cell from bright field images, which is kind of tells you that there is information hidden in that. And this is called, there are multiple names, but one of the names is in silico labeling. And in principle, if it works, what we can do is now train different models for each organelle, and then have integrative representation of a cell, which would be very cool. There were already multiple follow-up studies that showed that the proof of principle. In my lab, we are thinking of ideas on using in silico labeling for actually doing some discovering new biology. But the first part is actually understanding how it works. How it works, when it doesn't work. So actually doing the dirty work and trying to understand the limitation and how the network learns that. So it's really, nothing is published and it's all preliminary results. But what a cool result I think is that we correlated a measure for location stereotypy. So how the in silico labeling is correlated, how the organelle is correlated to the 3D geometry of a cell. This is the x-axis. As in the y-axis, you can see the accuracy of the in silico labeling, and you can see a very nice pattern here. And what we're thinking that we understand right now is that here, the network focuses on cell architecture. So if we look at the intermediate layers of a network that did the in silico labeling, we can actually see that it starts to capture the geometry of the cell. While if we look here, in organelles with low location stereotypy but high in silico labeling, we think that it's local texture within, a local specific texture within the bright field, which the network learns to localize. But this is all preliminary, and there is nothing I wouldn't put my mortgage on that. Another idea that is maybe a little bit less direct interpretable, but still I think that it can fall within the same category, is we're trying to apply to measure organelle spatial relations or interactions within high content screening assays. So in principle, if you look at it, for example, you have 5 different channels, you image 5 different organelles, and what usually we try to do is to understand, okay, when you perturb a cell this way, you change the orange organelle from a rectangle to a star. And what we are trying to understand is whether the spatial relations between the different organelles have changed. So if I break my hand, you can see that my hand is broken. But if I break my hand and I throw it to the other side of the room, the spatial organization is going to be, it's going to be a more specific readout or a more sensitive readout. What we think from here, I'm not going to show you data, but we think that it's much more sensitive. So when this happens much more dramatically. It's more reproducible, more specific, and it's actually complementary in some aspects. So you can have some changes here without changing the organization, and some changes in the organization without changing the organelles. Yeah. So again, in a year I can give us a full talk about that. Okay. So that was the first part, and for the second part, I'm going to talk about a problem that I'm very interested, which is multicellular organization, multicellular synchronization, and the main question here is each cell is an individual. It has its own machinery, and it makes decisions based on noisy cues that it gets from its neighboring cells and from the environment. And the question how this signal at the single decision at the single cell level propagates in order to coordinate the group. And what we are trying to do is bridge the scales from the local cell level to the group. And basically, we're taking a data-driven approach. So we're representing, when we look to a dynamic, we are modeling that as a network. So we have the fluctuating dynamics, and this could be anything. I'll show you a few examples in the next slide. This could be anything, and from that, I want to measure how one cell influences the other. And once we have that, we can construct a network, and then we can follow how information propagates from the local to the global scale within the network, and try to understand this process. So I'm a completely dry computational lab, but it has many perks, I think. One of them that I have a general idea, and now I get to collaborate with all these wonderful biologists that have these fantastic systems, and each system can fit a specific part of the general agenda that we have. So for example, with Ayelet Lessman from Tel Aviv University, we are looking at fibroblast cells that are embedded within 3D matrices. And here what is cool is that the cells are communicating mechanically by pushing the gel. And you can see here an alignment of the fibers and densification and alignment of fibers. What is cooler, when you look in live imaging, you can see you can actually follow the communication. You can follow the communication signals that is propagating between the cells. You can actually measure how the environment changes. And with Mike Overholzer from Kating Sloan Cancer Center, we're looking at collective cell death. So what is cool about that is that this is a very simple network. Once the cell die, then there is no feedback. So you can look at it as a network that is a tree, cycleless networks. So this is a simple network, and we can explore that. And for the rest of the time that I have, I'm going to talk about collective synchronization, which is a collaboration with Bosan from Oregon State University, and the student in my lab that is working on it is Amos Amir. Okay, so here's the system. So the system is a monolayer of endothelial cells, and the imaging here is calcium signaling. Endothelial cells are at the barriers of the blood vessel, and they need to be able to adapt to varying changes within the blood flow. For example, in acute wounds. What Bosan did, he developed a microfluidic device that allowed him to control very precisely the flows that the cells experience, and he can show that changing the shear stress that the cells experience also is correlating with the change in their calcium signaling. So this is a well-controlled, fantastic system to study how synchronization emerges within monolayers of endothelial cells. The experiment that Bosan did is applying multiple rounds periodical shear stresses. So what it gives us a different it gives us multiple cycles. We can treat each cycle as independent experiment, and then we can also follow how each cell is responding over time and changing its role in this communication network over time. So here is what we get. This is the calcium dynamics. In black here, you can see very nicely the cycles. This is the average. And in green, you can see the variability within the derivative of the calcium signal within the cells. So you can see that the system is synchronizing. At the beginning, there is huge variability between the cells, and over time, it converges, and the variability is reduced a lot. So the system is synchronizing over time. And this is a local property. So when we inhibit gap junctions, the system doesn't synchronize. So it's not that each cell is responding, is learning gradually to adapt to the external stress, but the cells need the gap junctions to communicate. If we do experiments with low density, also we get something similar. We need the cells actually communicating through the gap junction. So it is a local mechanism of communication that allows the system to synchronize. Okay, so as I told you, we're using here, we're trying to measure how one cell influences the other. And this is an asymmetric inherently, because one cell influences the other differently than the other cell influences the first cell. And what we use in order to do that is a Granger causality. Basically, we try to see if we try to predict the future of one time series based on its previous values, and then we see if we add information from the past of another time series, whether it allows us to improve the prediction. And this, and if we can do that, if it improves the predictions, then we can say that the cell A influences cell B. Once we do that, we look at the two measures for each single cell. One is the transmission score, how well a cell influences its local neighborhood, and the other is the receiver score, how it is influenced by other cells within the environment. So these are the two measurements that we look at. And when you look at the receiver and transmitter score over time, you can see first you can see spatial heterogeneity. So there are no clusters of very good communicating cells. And second, you can see that over time the system becomes more, there is more communication going in within the network. So there are more edges within this network. I'm not going to show you, but we show that the heterogeneity within this network is correlated with improved synchronization on the one hand. And when you look locally, homogeneity is actually good. So the cells need, when the cells are more similar to cells next to them, but there is a global heterogeneity within the multicellular network, this is the settings that are correlating best with the synchronization. Okay, the next question is, okay, but what is it something stochastic, or is it something inherent within the cell state? So does a cell always stays within the same state within the multicellular network? So what we did here, we correlated the transmitter and the receiver score over time of each single cell. And you can see here two things. First, you can see there is correlation. The correlation is positive. And second, you can see that the correlation increases, which means first that the cells have some memory of their previous cycle, their communication properties within the previous cycle. And second, that it is reinforced over time. The role in the communication network is reinforced all the time. Okay, so let's look at the single cells. So what we did here, we normalized the receiver and transmitter score, and then we looked, we defined 5 different regions within this transmitter receiver score spaces, and we defined 5 different roles. Individual cells are cells that are, they are not very friendly, they don't talk with anyone. And then we have the common, which are normal cells. We have leaders, which are cells that are very good at influencing other cells. Followers, they are influenced by other cells, and communication hubs. These are the most friendly cells that are good at transmitting information and at receiving information. If we look at it over time, we can see that, well, it's not surprising. I show you that the information flow grows, so it goes into the direction of the more communicating roles. And if we can look at it specifically, we can see that over time, the roles of the common and individual decreases, while the roles of a follower, leader, all the roles that are involved in communication increases. Yeah. So how much time do I have? 10 minutes? Oh, I have a lot of time. Including the question? Okay, so I don't have a lot of time. Okay, so I'll go really quickly because I was okay. Okay, so when we looked at transition between different states, and what you see here is only enriched transition. You can see here the memories. These are the self edges, and you can see a transition from left to right. So the cells are moving from less communicating to more communicating roles, and of course you can see this very spectacular enrichment in memory and communication hubs. So once cells reach the role of the communication hub, they tend to stay there much more than any other role. Okay, so basically what we hope to achieve here, what we wanted to show, is that the cells start by communicating locally within their local environment, and then over time they start to learn the local environment, and the information propagates from the local to the global scale. So we thought of, I think, a nice way of doing some analysis to show that, and I want to show you that now. So instead of looking at edges just between nearest neighbors, or just between close neighbors. We wanted to correlate the probability of having a Granger causality edge to the topological distance. So you can think about two scenarios. If you can think about two scenarios, when you see a negative correlation, so cells that are closer to one another have higher probability of talking with one another. This suggests a local mechanism that cells are talking with each other locally. What you see here, if we see something like this means that there is a positive correlation between the topological distance and the edge probability. So cells that are further apart are actually have a higher probability of an edge. This would mean that there is a global communication. Now, this could happen only if the cells that are closer to one another are already quite synchronized. Because once they are synchronized, we can basically they're already synchronized. There is no we cannot measure cause-effect relations here anymore. So here is what we got. These are the experimental results. On the x-axis, you can see the cycles. On the y-axis, you can see the correlation. And you can see the correlation actually strikingly switching from a negative correlation, so where the close cells are better at communicating than further cell, and then it switches through 0 to a positive correlation, where where cells that are further apart have a higher probability of having an edge. So we think that we identified that heterogeneity within the communication network, memory, and information flow contribute to collective information processing. We think that the cells start by communicating locally and stabilizing the local network, and then the information gradually propagates from the local to the global scale. This is driven by these communication hub cells, which are I didn't show you all the data, but these are pretty special group of cells. We have no idea anything about them yet, what is special about them. But we think that they are a part of the reason why the information propagates within this network so nicely. Okay, so these were two examples. The melanoma example, where we actually could eventually see something. And here in the calcium synchronization, I just showed you a movie at the beginning. There is nothing we can see with our eyes. We need to be careful with the conclusions, with what we think we learned here, but all that we learn here come from the numbers. Before I finish, I want to make one self-promotion. So I'm teaching a course on data science in cell imaging at BGU. It's a graduate level course, and you can find all the material, all the slides and everything available. And now it's also uploaded to YouTube, so you can hear me talk bad English and talk about data science in cell imaging. And with that, I'll finish. I still have some time for questions, I hope.
If you have any other questions regarding the second or even the first part of the talk, then please raise your hand.
Yeah, thank you very much for all the talk, and also for the non-deep learning last part. It's refreshing also on a deep learning day. We had colleagues so you mentioned causality, Granger causality. So I had some colleagues who work on this. I can't remember exactly the sample, but his answer was that he didn't have enough recording so that it was meaningful. So do you have any idea of how long should be your recording so that you can claim you have a time-oriented signal? The uncertainty of this measure of Granger causality?
Yeah, so eventually the readout is a statistical test, right? I mean, first you need to make sure that your data is stationary. You have some conditions that the time series need to fill, but in principle, eventually the output is a statistical test. So if you don't have a long enough time series, it will just not work, or will be too noisy to capture something. I think that eventually you see data that is very consistent, and I mean this figure is just amazing. I think I'm sorry, I have to look at it again. I mean, you don't get this type of things in experimental data. I mean, it's right something like this, right? A transition like this. So I don't know. I mean, yeah. but in principle, so it just would add noise to your network. You'll have less confidence in what you get.
Thank you. So what was the order of magnitude of your recording, the length of your recording here?
It was 2 frames per second, and it was for, I think, 200 minutes or 400 minutes or yeah, I think time. Yeah, but it is the time series are not 10 time points, right? It's hundreds of times.
Thank you.
Anyone has another question? Okay. Well, maybe we shall thank you and yeah.
So just before closing the session, we would like to thanks a lot again all the speakers from this session and from the other sessions from the module, and just maybe give you some key message from these modules. I don't know if Daniel is here, Daniel Sage. I haven't seen you. Okay, so Daniel is basically organizing the parcours thematic in deep learning. So please have a look in the program. So there will be some one table. In addition, there is some workshops which are specifically targeted to deep learning. So it's on the program. And maybe
yeah, perhaps a few words on what we might have learned today. No, so so in the, of course, in the fundamental course, which was given by David, and then in the different lessons. And so first thing is perhaps the application domains of deep learning. So we have seen really a number of applications, but what has been relatively clear is that image restoration is really a huge application domain. Segmentation, detection, classification. What is not very much, I mean, where there's relatively little in deep learning still is in registration and tracking. Of course, there are solutions, and Perrine mentioned some, at least for registration, but there I would say deep learning is not the predominant method yet. So, but whenever you have a problem in image restoration or detection or classification, and clearly I would say deep learning has become really the method of choice. So this has become, was relatively clear, and it has been confirmed again today by all the talks. And so maybe a few words on, because this was also when we discussed a little bit, deep learning really comes from a practical point of view with a change in paradigm. So before, what we did, and Daniel actually said it very nicely today, so we had these model-based approaches. So we worked on a couple of images. We tried to figure out what a nice model and a nice algorithm would be to extract this or that, or to do this or that analysis. And then in the end, when we were happy on our few images, then we actually applied it to the larger image set we wanted to analyze. So this was really the traditional way in which we kind of tried to solve bioimage analysis tasks. And this is clearly very, very different in deep learning. In deep learning, you first start by collecting a lot of data. You cannot start by working on a couple of images. So you really have to be systematic in the acquisition and in the annotation of data in order to make things work. And the second really important change in paradigm is really that it's focused on the data. So finally, the network that you're going to apply to data is never just the architecture of the method. It is always the architecture and the data that has been used to train it. And so this shift in focus was very important. And then if we just look at the lectures, I mean, there has been an overwhelming variety of things that we have seen. And so we do not want to repeat that now, of course. But just to think about what we have seen this morning, Daniel gave an overview over different tools, and he was very much concentrating also on pitfalls and problems and warnings, and all of this is very important because there has been a lot of enthusiasm with deep learning, but it has to be also a little bit, well, I say moderated or modulated by a realistic view of the assessment and the applicability of methods. Then, so Perrine talked a lot about image registration with and without deep learning, as we have managed, as I've just said before. Image registration is perhaps not the major application field for deep learning so far. Loïc had a very nice demonstration of self-supervised approaches among different things, but then he has talked about the classification of protein localization patterns, for instance. Martin has talked a lot about nucleus segmentation, Martin Weigert, and denoising. Diana has talked, focused mostly on learning with few samples. Sure, this was more in the medical domain, but I can tell you that in principle we have very similar problems in bioimaging too, and many of the methods that she has actually been showing would be, to some extent at least, also applicable to biological imaging. And then Asafi has really talked about applications of explainable AI, and then mostly with applications to phenotyping and so forth, more downstream analysis, so getting away from segmentation and denoising and more moving then really to inferring biological function by the analysis of artificial intelligence. And so, yeah, so this is what we wanted to summarize a little bit. Now what is really important, of course, for deep learning as for any other method, nobody should actually just apply these methods without having a basic understanding of what is required, what are the basic requirements of these methods. And I think we have really seen today a large panorama of theoretical concepts and then also practical applications. And I think the speakers always have managed it very nicely to give you an idea of really what the requirements actually were to get to these very fantastic results, right? So it's important to understand all of these requirements. But conversely, it's also not very useful just to know something about requirements and theoretical aspects without having the practical aspects too. And these practical aspects, they will be covered now in the ateliers. And perhaps you want to say something about the parcours.
Okay, yeah. Unfortunately, everything is sold out, but you can take your chance, because some people get tired over the week. So if you are really interested, you can join even if you were not fully registered. So we have different ateliers. So there is some without any coding, using codes, using tools where you can fully develop a deep learning architecture without coding, that is under NIME. We have also stuff under the Nikon software, which is more press button, but we always with the keeping in mind to try to understand what we do. And then there's a large panel. So Daniel is there with deep imaging as a demo, and I encourage you also to go to the hands-on on deep learning zero mic cost, not in the right order, but you have also a demo on this. Plus other more applied hands-on on specific samples, so for spheroids. And there is another one, I can't remember exactly, but it's another type of samples also. And plus, don't forget, so on the first day, we have a final discussion. That'd be nice to have your feedback from your understanding of this week, for also your test on the during the hands-on, and for possible more training provided by the GDR. So please also be part of the table ronde on Thursday.
So thank you, and just the last word, maybe also to thank our chairman and chairwoman of the day, so Sofia Imperato from ISPCI, Alan van der Berg, NIFNIL, and Marine Boyer from Diffusion. So thank you for sharing. And thanks for all the speakers as well.